Today we're going to be talking about the female parts of a flower, called the carpel or pistil. So let's go ahead and draw a diagram of this. So up here, this is going to be a carpel or pistil. And the very top part of the carpel is referred to as the stigma. And stigma means sticky spot. And we want that to be sticky because the pollen actually have to be caught or stuck to the stigma. The next part is going to be the style. And the large part, probably the most important part, is going to be the ovary. Now, inside the ovary are some very important parts. Let's go ahead and shade in the ovary, or the wall of the ovary. So I'm shading in the wall of the ovary. Now, inside this ovary is going to be an ovule. Usually many ovules, but I'm just going to draw one for clarity. So we're going to go ahead and shade in the wall of the ovule. So we'll go ahead and label that ovule. And there's going to be a little hole here in the ovule. This hole has a special name. The hole here is referred to as the micropyle. And this micropyle is the hole through which the pollen tube is going to enter the ovule. Now, let's just take a look at a piece of pollen real quick, a grain of pollen. Here's a grain of pollen. Go ahead and label that. That's going to be a pollen grain. And once this pollen grain is stuck to the stigma, it begins to germinate or grow. And it actually grows a tube called a pollen tube. And at the tip of that pollen tube are going to ride two non-flagellated sperm. And those two non-flagellated sperm are going to ride in that pollen tube right to the egg. They're going to get delivered right to the egg, right where they're needed. Now, inside the ovule are going to be 7 cells. And these 7 cells collectively are going to be referred to as the embryo sac. So let's go ahead and draw these 7 cells. There's going to be 3 at the very opposite end or back. So 1, 2, 3. Each one has a single nucleus. These are referred to as antipodal cells. And they're not going to be that important in the talk I'm giving right now, but I just wanted to make a note. Those are antipodal cells. And we've got 3 other cells, 1, 2, 3 right here. And again, each one has a single nucleus. And the cell right here in the middle, right where the micropyle is, that is referred to as the egg. And these nearby cells, one on either side, are called synergids. And these cells are going to be important, although you're not going to have to remember the name synergids. But these cells actually produce chemicals and actually create a concentration gradient. And the pollen tube is actually going to follow that concentration gradient right to the egg. So these synergids are very important. They produce that concentration gradient that allows the pollen tube to deliver those sperm right to the correct location. And our last cell, which is our large central cell, consists of 2 nuclei. And these 2 nuclei are oftentimes referred to as polar nuclei. So let's go ahead and label that. That's the central cell. Now again, collectively, all these green cells, these 7 cells, are referred to as the embryo sac. That's going to be the female gametophyte in angiosperms. And it consists of 7 cells. And we need to really remember 2 of those 7 cells. One being the egg, which is N, and the other one is the central cell, which is 2N. Now, let's go ahead and talk about what's going to happen when fertilization takes place, once pollination takes place. When a grain of pollen lands on the sticky stigma, it's going to produce a pollen tube. And this pollen tube is going to grow. And it actually releases digestive enzymes that eat a hole right through the style, right through the ovary. And we're going to follow that concentration gradient that we talked about earlier. And this pollen tube is going to grow right around through the ovary, right through the wall of the ovary, and it's going to deliver its cargo, 2 non-flagellated sperm, right through the micropyle and right to the egg. So one of those sperm are going to go and they're going to fertilize the egg, and that's ultimately going to produce an embryo. And the other sperm is going to go and it's going to fertilize this central cell. And the central cell ultimately is going to become the endosperm, the food for the future embryo. So let's go ahead and write these things down. The central cell eventually is going to turn into the endosperm after fertilization. And that's going to be the food for the future embryo. And the egg ultimately is going to become our embryo. Now, once seed development occurs, once that begins to happen, we're going to have some hormones start to be produced, and the ovule is actually going to begin to mature, and it's going to mature into the seed coat. And the seed coat is going to offer protection to the seed. And the ovary is going to further mature or ripen into the actual fruit. So the ovary is going to become the future fruit. And ultimately, that fruit is going to help disperse this seed that we're producing. If it's a fleshy fruit, oftentimes it attracts animals, which then eat it. The seed doesn't get digested, and it usually gets released later on. Or possibly the seed could be like a burr and would stick to an animal's fur, and then that animal would eventually deposit that seed in another location. And that's going to be it for the parts of a carpel.